Okay, folks. So I just wanted to go ahead and go over what to expect for Unknown Lab 1. So with Unknown Lab 1, what we do is we start our series of 3 labs where we take an unknown culture, which I'll give you, and you'll go through the 3 labs, the first one being our staining procedures, the second one, which is similar to Lab 6, and the third one, which is similar to Lab 8. And as you go through the different procedures and the different media, you'll learn more and more about your unknown organism and eventually be able to identify it. So in Unknown Lab 1, we will actually complete the staining procedures that we learned in the class. So we'll take our unknown organism and we'll do a simple stain, a gram stain, and an endospore stain. Okay? And so from those staining procedures, we should learn its shape and arrangement, whether it's gram positive or gram negative, and whether or not it's producing endospores. Okay? But we will also inoculate the media for Unknown Lab 2. And Unknown Lab 2 looks very similar to Lab 6. So it's your MSA, MAC, PEA, and fluid thioglycolate. And so in that way, as we inoculate our media in the first day, on the first day, then on the second day, we'll be able to go through and actually read that media. Okay, so make sure to kind of manage your time appropriately for this lab. You'll want to start your 3 slides at the same time with your organism, your unknown organism on there, and you're going to go through the simple stain procedure, gram stain procedure, and endospore stain procedure. Now, it's likely that the simple stain is done first, the gram stain is done second, and then since the endospore stain is the longest procedure, that'll likely be done third. And so you can just go through and do your microscopy and look at your organism as you go through the different procedures, okay? But also make sure to time in your inoculation so that you get your media, your MAC, MSA, PEA, and thioglycolate media inoculated as well with your unknown organism, okay? So let's go ahead and review the 3 staining procedures which you'll be doing in the first lab, okay? So remember, for the staining procedures, we have a series of steps. We always have a primary stain. If it's a simple stain, that's all we have. But if it's a complex stain like the gram stain or the endospore stain, then we also have a mordant, some sort of decolorizer, and a secondary stain or a counter stain. Okay, so let's just go through these 3 different procedures: simple stain, gram stain, an endospore stain, and just go through the different steps for each. Okay, so for the simple stain, we could use many different simple stains for this procedure, but in this specific class, we use crystal violet. Okay. And so since this is a simple stain rather than a complex stain, so these are complex stains. Okay. But this is a simple stain, so we don't have a mordant, a decolorizer, or a secondary stain. We just put on our crystal violet and then rinse it off, blot dry, and then that is ready to look at under the scope. Okay? For our gram stain, the gram stain is the first of the 3 that's a complex stain, so we'll actually go through these 4 steps. So for our primary stain, we're going to have also crystal violet. Okay? And that's going to be our primary stain. Then we have our mordant. The mordant in this case is iodine. Iodine's going to bind that crystal violet into the peptidoglycan layer of our organisms. Then we have a decolorizer. And that's alcohol. We have a pretty harsh decolorizer for the gram stain because we actually want to wash out the crystal violet from the gram-negative organism. So at this point, gram-positive organisms will be purple, but gram-negative organisms should have been decolorized and should be transparent. So we come in with safranin, which is going to stain everything, but it's going to allow us to differentiate gram-negatives as pink and gram-positives as purple because they retain the stain from these first two steps. Okay, so that's our gram stain procedure. At the end of the gram stain procedure, we should be able to differentiate the cell envelope anatomy and know whether our organisms are gram negative or gram positive. Okay. Okay. Then our third staining procedure is the endospore procedure, staining procedure. And the first or the primary stain in the endospore staining procedure is malachite green. Okay, malachite green. Our mordant in this case is not a chemical mordant. It is the combination of heat and time. So you have an extended heat fix and an extended stain with the malachite green being on there for 15 to 20 minutes. Okay. And then our decolorizer is actually a moderate and a soft decolorizer. That's water. So at this point, after we've added the malachite green after an extended heat fix and for an extended period of time, we're hoping that if endospores are present, they pick up some of that stain. And then we'll rinse the excess stain out, but hopefully not whatever's been picked up from the endospore. And then we need to come in with a counter stain, which again is going to be safranin, so that we can actually see the vegetative cells around the endospores. So since this is a complex staining procedure, it allows us to differentiate whether organisms are or are not producing endospores. So whether they're not are or are not producing endospores at that given time, okay? And so in this way, for Unknown Lab 1, you'll learn about your organism, your unknown organism. You'll be able to see its shape and arrangement in the simple stain, which you will also be able to see in the other stains. But then in the gram stain, you'll also know whether it's gram positive or gram negative. And in the endospore stain, you'll know whether it does or does not produce endospores. Excuse me. Just got a phone call there. But that will allow you to go through the first part of that Appendix B, which are those tables that go through all of the expected results for each of the potential unknowns and start to narrow down. Okay, is my organism bacillus shaped or coccus shaped? Is it gram positive or gram negative? Does it or does it not produce endospores? And then you'll start to actually narrow down which organisms yours might potentially be. You'll also do your inoculations for Unknown Lab 2 on MAC, MSA, PEA, and thioglycolate. And on those plates, just, get your organism on there. You can do streak inoculation, or you can basically either way, any way that you can get that organism onto the plate, and that will allow you to then read your results for Unknown Lab 2 when we meet for the second Unknown Lab. Okay, so let me know if you have any questions about Unknown Lab 1, and that'll get us started in identifying our unknown organism, which is just that final project in the lab for microbiology.